A chromosome abnormality, disorder, anomaly, aberration, or mutation is a missing, extra, or irregular portion of chromosomal DNA. It can be from an atypical number of chromosomes or a structural abnormality in one or more chromosomes. Chromosome mutation was formerly used in a strict sense to mean a change in a chromosomal segment, involving more than one gene. A karyotype refers to a full set of chromosomes from an individual that can be compared to a normal karyotype for the species via genetic testing. A chromosome anomaly may be detected or confirmed in this manner. Chromosome anomalies usually occur when there is an error in cell division following meiosis or mitosis. There are many types of chromosome anomalies. They can be organized into two basic groups: numerical and structural anomalies. Numerical disorders. This is called aneuploidy, an abnormal number of chromosomes, and occurs when an individual either is missing a chromosome from a pair, monosomy, or has more than two chromosomes of a pair, trisomy, tetrasomy, etc. In humans, an example of a condition caused by a numerical anomaly is Down syndrome, also known as trisomy 21. An individual with Down syndrome has 3 copies of chromosome 21, rather than 2. The frequency of trisomy 21 has been determined to be a function of advanced maternal age. An example of monosomy is Turner syndrome, where the individual is born with only one sex chromosome, an X. Sperm aneuploidy. Exposure of males to certain lifestyle, environmental and/or occupational hazards may increase the risk of aneuploid spermatozoa. In particular, risk of aneuploidy is increased by tobacco smoking, and occupational exposure to benzene, insecticides, and perfluorinated compounds. Increased aneuploidy is often associated with increased DNA damage in spermatozoa. Structural abnormalities. When the chromosome structure is altered, this can take several forms. Deletions. A portion of the chromosome is missing or deleted. Known disorders in humans include Wolff-Hirschhorn syndrome, which is caused by partial deletion of the short arm of chromosome 4, and Jacobson syndrome, also called the terminal 11q deletion disorder. Duplications. A portion of the chromosome is duplicated, resulting in extra genetic material. Known human disorders include Charcot-Marie-Tooth disease type 1A, which may be caused by duplication of the gene encoding peripheral myelin protein 22, PMP22, on chromosome 17. Translocations. A portion of one chromosome is transferred to another chromosome. There are 2 main types of translocations. Reciprocal translocation. Segments from 2 different chromosomes have been exchanged. Robertsonian translocation. An entire chromosome has attached to another at the centromere. In humans these only occur with chromosomes 13, 14, 15, 21, and 22. Inversions. A portion of the chromosome has broken off, turned upside down, and reattached, therefore the genetic material is inverted. Insertions. A portion of one chromosome has been deleted from its normal place and inserted into another chromosome. Rings. A portion of a chromosome has broken off and formed a circle or ring. This can happen with or without loss of genetic material. Isochromosome. Formed by the mirror image copy of a chromosome segment including the centromere. Chromosome instability syndromes are a group of disorders characterized by chromosomal instability and breakage. They often lead to an increased tendency to develop certain types of malignancies. Inheritance. Most chromosome abnormalities occur as an accident in the egg cell or sperm, and therefore the anomaly is present in every cell of the body. Some anomalies, however, can happen after conception, resulting in mosaicism, where some cells have the anomaly and some do not. Chromosome anomalies can be inherited from a parent or be de novo. This is why chromosome studies are often performed on parents when a child is found to have an anomaly. If the parents do not possess the abnormality, it was not initially inherited, however it may be transmitted to subsequent generations. Acquired chromosome abnormalities. Most cancers, if not all, could cause chromosome abnormalities, with either the formation of hybrid genes and fusion proteins, deregulation of genes and overexpression of proteins, or loss of tumor suppressor genes. See the Middleman database and the Atlas of Genetics and Cytogenetics in Oncology and Hematology. Furthermore, certain consistent chromosomal abnormalities can turn normal cells into leukemic cells, such as the translocation of a gene, resulting in its inappropriate expression. Detection. Depending on information we want to obtain, different techniques and samples are needed. For the prenatal diagnosis of a fetus, amniocentesis, chorionic villus sampling or circulating fetal cells would be collected and analyzed in order to detect possible chromosomal abnormalities. For the pre-implantational diagnosis of an embryo, a blastocyst biopsy would be performed. For a lymphoma or leukemia screening, the technique used would be a bone marrow biopsy.
Please subscribe and thanks for watching.